Tell.
Welcome to the Deep Dive. Today we're diving into a really pressing challenge in cancer treatment, triple-negative breast cancer, or TNBC.
Right. It's known for being particularly aggressive.
Yeah, and unlike many other breast cancers, we don't have those specific receptor targets. You know, the ones we can go after with payload therapies.
Exactly. Which often leaves chemotherapy as the main option.
And chemotherapy, as you know, it can be tough. Really tough on the body.
Absolutely.
And, thinking bigger picture, cancer is the second leading cause of death in the U.S.
A staggering statistic.
It really is. And they project the number of survivors will top, 22 million soon? So the need for better options is just crystal clear.
Especially for TNBC.
Right, especially because it disproportionately hits younger African-American and Hispanic women, particularly those carrying BRCA1 mutations. It's a serious concern.
It really does highlight the urgent need for more targeted strategies, doesn't it?
It does. And the research we're exploring today, well, it offers a really promising direction, I think.
A very promising one, yes.
Exactly. So our mission in this deep dive is to really unpack a recent research article. It details a potential breakthrough, the discovery of new drug candidates for TNBC.
And they focused on a specific protein, PARP1.
PRP1, right. Okay, so let's break this down a bit. PRP1 is a key player in how our cells repair damage to their DNA.
It's vital for that process.
Now, there's this fascinating concept called synthetic lethality. Have you heard about this?
Oh, yes. It's a really clever approach in cancer therapy.
So imagine cancer cells,
Yeah.
right? They already have a weakness, maybe in their DNA repair, like those with BRCA1 or BRCA2 mutations.
Okay, so one pathway is already compromised.
Exactly. Then, if you inhibit another crucial repair pathway like the one involving PRP1, you basically overload the cancer cells.
Too much DNA damage accumulates.
Right. They just can't handle it and they die. It's like removing the backup generator when the main power is already flickering.
A great analogy. And it gets even more intricate with PRP inhibitors. It's not just about blocking the enzyme.
How so?
Well, some of these inhibitors can actually get trapped on the DNA molecule itself.
Trapped.
Yeah, think of it like literally gumming up the works. It physically blocks the repair machinery.
Wow, like throwing a wrench in the gears.
Precisely. That trapping effect can make these inhibitors even more potent, significantly amplifying their cell-killing ability.
That's powerful. Okay, so the article we're digging into today looks at how scientists found potential new PRP1 inhibitors for TNBC.
Using a really modern approach, structure-based, virtual, high-throughput screening.
Right, a virtual screen. And they started big, didn't they, with a huge library of molecules.
Huge is right. 4.59 million compounds. All sourced from the ZINC database, which contains compounds considered lead-like.
Meaning they have properties that make them good starting points for drug development.
Correct. And the beauty is, they did this initial screening computationally.
So, not in a wet lab, pipetting millions of tiny samples.
Exactly. Imagine the time and cost saving. They used computers to simulate how each molecule might interact with PRP1.
Okay, tell us more about that virtual screening. How did it actually work?
Well, they used computer models to predict which of those millions of compounds might bind strongly to PRP1 and potentially block its activity.
They used a specific tool, right, of ERTRED?
Yes, FREED for the molecular docking. And they needed a map of the target, the PRP1 protein.
The 3D structure.
Exactly. They use the crystal structure from the protein databank, PDB-ID5800. It gives you a really detailed picture.
And this map highlights the important spots.
Crucially, the active site. That's where the enzyme does its work. And specific amino acids within that site, like GLU988, TR896, ASP899, TR907.
Like the critical parts of the lock they're trying to pick.
Precisely. The virtual screening looks for compounds, the keys, that fit well and interact strongly with these key amino acids.
So millions of virtual keys tried in the PRP1 lock. The better the fit, the higher the chance it's an inhibitor.
That's the core idea, yes. Predicting binding affinity.
Okay, but 4.59 million is still way too many to test physically.
Yeah.
How did they narrow it down?
Right, that's where the filtering comes in. It's a crucial step. After that initial docking, they got about 16,000 potential hits.
16,000 is better than millions, but still a lot.
Still far too many for efficient lab testing. So they applied more filters, They looked at the docking score, of course, but also things like shape complementarity, how well the compound's shape matches the binding site.
Makes sense.
And importantly, those lead-like properties we mentioned earlier, they wanted compounds suitable for further development.
Lead-like properties. That sounds critical. What does that actually mean in practice?
Oh, absolutely vital. If you want a compound to ever become a real drug, they assess AD-MET properties.
That's in MET. Absorption, distribution, metabolism, excretion, and toxicity.
You got it. These predict how a drug might behave in the body. Will it get absorbed? Where will it go? How is it broken down? Is it safe?
Key questions.
Definitely. They also use drug likeness rules. You might have heard of Lipinski's Rule of 5.
Yes, I have. It looks at things like size, fat solubility.
Exactly. Molecular weight, lipophilicity, polarity, hydrogen bonding potential. These rules help predict if a compound is likely to be orally available, for instance.
And they use one more filter, right? Something about false positives.
Yes, the Payne's filter. Panis interference compounds. Some molecules can look like hits in lab tests, but they're actually just messing with the assay itself.
So it weeds out the tricksters.
That's a good way to put it. It avoids wasting time and resources on misleading results.
So after all this virtual sifting and sorting, this multi-layered filtering, they got down to a manageable number.
Precisely. From the initial millions through the 16,000 hits down to a very select group of just 25 top candidates.
25. That feels much more achievable for lab work.
Absolutely. These were the ones that looked most promising based on all the computational analysis.
And then it was finally time. Take these virtual leads into the real world, into the lab. How did they test them?
They used a standard method called a PARP1 colorimetric assay.
Okay, what does that measure?
It's a biochemical test. It measures how well a compound blocks PRP1's enzymatic activity. Specifically, it tracks the buildup of a molecule called PR-poly, ADP ribose.
PR is the product of PRP1 doing its job.
Exactly. So if your compound is inhibiting PRP1, you'll see less PR being produced.
Makes sense. Less product means the enzyme is blocked.
Correct. And to be sure the results were reliable, they did the assay multiple times for each compound in triplicate.
Good scientific practice. So, the moment of truth. What did they find? Did any of those 25 show real promise in the lab?
Yes, the results were definitely encouraging, very promising, in fact.
Tell me more.
So out of the 25 tested, 4 compounds showed strong inhibitory activity against PRP1, really knocked down its function.
4 strong hits. That's pretty good from 25.
It is. And another 12 showed moderate inhibition, so a decent hit rate overall from the virtual screening.
Interesting. Did they notice anything about the structure of the strong ones?
Yes. 2 of the most potent ones, they called them hit 10 and hit 22, were structurally related. They're benzopyrazole analogs.
Okay. What's significant about that structure?
Well, the researchers noted the structure has features good for binding PRP1, an amide group for hydrogen bonding, which is like a specific sticky point,
Right.
and an aromatic ring, which can participate in something called pus stacking. Think of it like flat parts of molecules sticking together.
So features that help it dock tightly into the protein.
Exactly. It helps it bind effectively.
And they really zoomed in on one of these hits, hit 22, didn't they?
They did. Hit 22, which is ZINC0000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000 Well, they looked closely at how it interacted with PRP1. They found specific contacts ionic interactions with an amino acid called PASP.
Okay.
And those pos stacking interactions we just talked about, specifically with Ty907, these precise interactions explain its strong binding.
And they looked at its properties too, like the AD-MET stuff.
Yes, they characterize its molecular properties. Its weight is good for a drug 333.39 gmol, not too large. It has 5 rotatable bonds, giving it some flexibility. Good numbers for hydrogen bond acceptors and donors.
All sounding positive.
Right. Favorable values for molar refractivity, polar surface area, and a log p-value of 2.93, suggesting it should be able to cross cell membranes reasonably well.
And the binding strength, did they quantify that?
Their simulations predicted a very high binding affinity score of 19.6. Really strong predicted binding.
Okay, that's the prediction. What about the actual lab test, the IC50?
That's the key experimental validation. The IC50 tells you how much drug you need to inhibit 50% of the enzyme activity. Lower is better, more potent.
So what were the numbers for the top 4?
Okay, so for hit 17, the IC50 was 5.3518 micromolar. For hit 10, it was 6.3031 micromolar. Hit 24 was 7.4533 micromolar.
And hit 22.
Hit 22 came in at 1.4767 micromolar.
Wow, significant. That's quite a difference. And you mentioned something about its structure being novel.
That's another really interesting point. The chemical structure of hit 22 contains what's called an indole pyrimidine moiety.
And that's unusual.
According to the researchers, yes. This specific structural feature hadn't been previously identified in known PARP1 inhibitors. So it represents a potentially new class of inhibitors.
A novel scaffold. That's always exciting in drug discovery.
Absolutely. And it also lined up well with some of their earlier work using molecular dynamics simulations. A similar compound, Z04, was predicted to be effective, and hit 22 confirmed that potential experimentally.
So the computation and the experiment agreed. That must have been satisfying.
Always good when that happens. It builds confidence in the approach.
Okay, so they've got this potent, novel inhibitor. But what about safety? You can have the best inhibitor in the world, but if it's toxic...
It's a non-starter, exactly.
Yeah.
So those 80 MET predictions become crucial again at this stage.
What did they find for the top 4 hits?
Well, first off, all 4 passed the general drug likeness criteria. That's good.
And the pains filter.
None of them triggered pains alerts, which is also reassuring. Suggests their activity is likely real, not an artifact.
Okay. What about actual toxicity predictions?
They were all generally classified as class 4 toxicity in one system, but the specifics varied. Importantly, hits 17, 10, and 22 were predicted to have low potential for liver toxicity.
That's a big one. Liver toxicity kills a lot of drug candidates.
It certainly does. Hit 24, however, was flagged as potentially hepatotoxic, and it also had the lowest LD50 value.
LD50, that's the dose that's lethal to 50% of test animals, right? So lower means more acutely toxic.
Correct, suggesting hit 24 might have a narrower safety margin.
What about other safety flags,
Yeah.
like causing cancer or general cell damage?
Good questions. For carcinogenicity, the predictions suggested hit 17 and hit 22 were not likely to be carcinogenic.
But hits 10 and 24 were flagged.
Yes, they were flagged as potentially carcinogenic based on the models. For general cytotoxicity broad cell damage, none of the 4 were predicted to be cytotoxic.
Okay, so putting it all together, hit 22 seems to be pulling ahead again.
It really does. When you look at the overall profile, potent inhibition, novel structure, good drug likeness, and the safety predictions, hit 22 looks the most favorable. No predicted liver toxicity, not carcinogenic, not cytotoxic, and the highest LD50 value of the group, 1100 milligiacoldam.
Suggesting a potentially better safety profile overall.
Based on these predictions, yes. It appears less harmful compared to the other top hits.
Okay, so if we really distill this whole deep dive down, what's the main takeaway message from this research?
I think the key conclusion is the successful identification of hit 22 ZOE0000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000000001 as a really promising lead compound.
A lead compound for PARP1 inhibition in TNBC.
Exactly. It hits the key points. Strong activity in the lab, a novel chemical structure, and crucially favorable predicted 80 MET properties, and a comparatively lower toxicity risk profile.
It really underscores, doesn't it, both the ongoing challenge of treating TNBC, but also how vital these innovative computational approaches are.
Absolutely. Computer-aided drug design like this virtual screening can massively speed up the discovery process. Finding leads like hit 22 would take much longer, be much more expensive otherwise.
It feels like a significant step forward, then, in this search for better, maybe kinder treatments for this tough disease.
I agree. Identifying a novel compound with this combination of potency and predicted safety is genuinely exciting. It opens up real avenues for future drug development work.
And could hopefully offer some new hope for patients facing TNBC down the line.
That's the ultimate goal, isn't it?
It certainly is. And it definitely leaves you thinking, what are those critical next steps? What further studies are absolutely essential now to see if hit 22 could actually make it to the clinic? That's something to ponder. Thanks for joining me for this deep dive.
Pharma Pal.